Good morning. So before I begin, allow me to take a moment to reflect on the context of this work. It was early 2020 when the world was in turmoil, borders were closing, hospitals were quite overwhelmed, and every day brought more uncertainties. As a pharmaceutical science PhD student in Thailand at that time, I found myself not just in a classroom or in the laboratory, but in the middle of a global crisis. So when the first wave of COVID-19 struck Asia, the Thai government quickly mobilized the scientific community. Almost all research in pharmaceutical sciences was redirected to support the pandemic response. From vaccine trials to antiviral development, even diagnostic tools and the public health solutions. And I was fortunate and deeply humbled to be part of these efforts. What began as a dissertation project became something far more urgent, a contribution to the fight against a virus that was devastating families and communities. As a young Filipino scientist working away from home, this experience reshaped my understanding of science. It wasn't just about publishing or finishing my degree or even my experimentation. It was about public service, impact, and solidarity. The research that I'm about to present was conducted during those challenging months, driven not only by scientific curiosity, but by the shared mission to save lives. It's a story of resilience, collaboration, and innovation under pressure. And with that, allow me to begin my presentation. Good day, everyone. I am Dr. Cathy Cruz, a postdoctoral fellow at Chulalongkorn University in Thailand and a faculty member at Our Lady of Fatima University in the Philippines. Today, I'll be sharing our research on enhancing the nasal delivery and antiviral effectiveness of favipiravir using mucoadhesive cytosine alginate nanoparticle, a study conducted during the Thailand's pandemic response with relevance for future antiviral strategies. So here's the outline of my presentation. I'll begin with a brief introduction and context, followed by our research objectives and hypothesis. Then I'll walk you through the methodology, key results, and discussion, and end with our main conclusion and the broader impact of this work. In early 2020, as cases surged globally, the repurposed antiviral drugs like the favipiravir became our beacon of hope. However, its oral formulation would require high doses of up to 1.9 grams per day, which came with risks to toxicity, especially in vulnerable population. And this urgent context is what drove us to develop a localized, safer delivery system. So why intranasal delivery of antiviral drugs? So during these times, we don't know much about the virus. But one of the earliest and most consistent findings during the COVID-19 was that the virus would enter and sheds heavily in the nasal pharynx and also the nasal cavity, even in asymptomatic individuals. And this makes the nasal route an ideal target for localized antiviral therapy, which should allow us to reroute the drug delivery directly to the infection site. However, there are some challenges. One, the epithelial barrier has a tight junctions, which would limit the absorption. And second, the mucosal layer, which renews every 30 minutes, would cause rapid drug clearance. So while the rerouting is promising, it would require a specialized delivery system that could both adhere to and penetrate through the mucus, which our mucoadhesive nanoparticles were designed to. At the peak of the pandemic, what we need is a fast, safe, and targeted delivery options, wherein the nasal route offered a strategic advantage, directly targeting the upper respiratory tract where the virus first enters. So this was especially critical in 2020 when systemic therapies were limited and vaccines were not yet available worldwide. Our muco-enhancing nanoparticles were designed to address two critical challenges in the nasal drug delivery. First is the mucociliary clearance, and the second is the rapid entrapment in the mucous layer, wherein conventional nanoparticles often get trapped into the mucus and are quickly cleared before reaching the epithelial surface, thus resulting in a poor drug absorption. To overcome this, we developed a stable nanocarrier, which are composed of alginate and chitosan as the polymers. The alginate is a natural biocompatible hydrophilic polymers that forms a protective gel matrix that is ideal for controlled drug encapsulation. While the chitosan is a cationic polymer that has mucoadhesiveness that would enhance the residence time by binding electrostatically to negatively charged mucus and opens tight junctions, thus promoting drug permeation through the nasal epithelium. So together, these combinations would enable the transmucosal delivery of bacteria, thus improving the deposition, prolonging the contact time, and also increasing the therapeutic efficiency at the site of infection. Our approach will provide three major advantages. First is virus infected. The overall objective of this study was to fabricate polymeric nanoparticles that could successfully deliver and deposit favipiravir in the nasal cavity and amplify its antiviral activity against SARS-CoV-2. First, we wanted to fabricate, optimize, and characterize the nanoparticle using box bending design and response surface methodology. Second is to evaluate the nasal permeation and biocompatibility using in vitro cytocompatibility and nasal histopathology. And the third is to assess the antiviral activity and cellular uptake using surrogate coronavirus model in viral cells. For this objective, we fabricated the favipiravir-loaded nanoparticles using emulsification, followed by ionotropic gelation combining alginate and chitosan. The optimized nanoparticle had a mean size of 233 nanometers, a negative surface charge, and a high drug loading and encapsulation efficiency. As shown in the SEM and TM images, the particles were spherical and well dispersed with uniform morphology, wherein the size distribution and the zeta potential plots further confirms the stability and the suitability of the formulation for intranasal delivery. The permeation study, we use porcine nasal mucosa and differential diffusion assays to simulate human nasal delivery. Our results showed that the mucoadhesive nanoparticle enhanced favipiravir permeation by 6 times and the epithelial deposition by 8 times compared to the free drug. And this is due to the chitosan's ability to open up epithelial tight junctions and its strong mucoadhesiveness and mucopenetrating properties, thus allowing better drug retention and absorption in the nasal cavity. Tissues exposed to the formulation showed no signs of apoptosis, necrosis, or inflammation. These results were reviewed by a blind pathologist confirming the biocompatibility of our nanoformulations. Before antiviral testing, we ensured that the nanoparticle formulation was non-toxic. In vitro tests on RPMIs, 2650 human nasal epithelial cells showed high cell viability of more than 80%, even at elevated nanoparticle concentration, thus indicates a strong cytocompatibility and minimal toxicity. During the height of the pandemic, the direct handling of SARS-CoV-2 virus was not feasible due to strict biosafety restriction. So in collaboration with the Faculty of Medicine at Chulalongkorn University, we use a surrogate model, which is the porcine epidemic diarrheal virus or the PEDV, which shares over 80% genetic similarity to its RNA polymerase with SARS-CoV-2, but poses no risk to humans and can be easily studied under lower biosafety conditions. For the assay, we use the very easy cells, which stably expressing EGFP, which were grown in confluence and then treated with our samples. So the PEG was engineered also to express M-cherry, which is a fluorescent protein that allows real-time quantification of viral replication. So the more the virus replicates, the more M-cherry signals can be detected, as seen as a golden yellow sensation under the imaging. So the total M-cherry intensity was then measured using harmony high content imaging, giving us an accurate and quantitative readouts of the antiviral activity. On the left is a quantitative summary of the M-cherry fluorescence intensity. So our formulation of the nanoparticle in blue showed significant viral inhibition starting at 1.44 micrograms per ml with an EC50 of 6.63 micrograms per ml. In contrast, the free favipiravir only became significantly effective at a concentration of more than 11.5 micrograms per ml, with an EC50 of more than 230 micrograms per ml. On the right are actual images of the Harmony High Content Imaging System. As you can clearly see, fewer M-cherry positive or the yellow sensation in the nanoparticle group, even at lower doses, indicating a suppressed viral activity. Thus, these findings would confirm that the nucleic acid nanoparticle could enhance the favipiravir antiviral efficiency by more than 35 times, likely due to the improved causal formation and also the cellular uptake. In conclusion, our formulation achieved four key outcomes. First, we developed a stable nanocarrier using tyrosine alginate, optimized for nasal mucosal delivery of favipiravir. Second, the system showed a strong mucoadhesive and mucopenetrating properties, resulting to a high drug deposition and permeation across the nasal epithelium. And the third, these enhanced the mucosal delivery, translated into increased cellular uptake, thus ensuring that more active drug reach its intracellular targets. And finally, this led to dramatic improvements in the antiviral activity, with our formulation showing over 35 times greater effective than the free favipiravir against coronavirus model. So altogether, this work offers a promising strategies for intranasal antiviral therapy, especially important in times of respiratory virus outbreaks. I'd like to close this segment by connecting our research to the real world impact. In 2022 of October, Thailand made headline as the first country in the world to approve the manufacture of COVID-19 nasal sprays, which is a major milestone in the pandemic response. And this innovation was built on the very same foundation of intranasal mucosal targeted therapy that we were working on during the peak of the pandemic. And it's a proud moment, not just as a scientist, but as someone who was part of the collaborative effort that turned crisis-driven research into real deployable health solution. Having worked on this research during one of the most critical global health emergencies in the recent history, I witnessed firsthand the power of timely and locally developed pharmaceutical solutions. And that experience drove my mission to bring these technologies back to the Philippines, not just to innovate, but to prepare for the future health threats that we may face. And with that, I extend my deepest thanks to my advisors, mentors, and collaborators at Chulalongkorn University. the NSTDA Thailand, and Our Lady of Fatima University, whose support made this research possible. And that's all. Thank you very much, everyone, and have a great day.